Assalamualaikum warahmatullahi wabarakatuh. We are from Group 1 ongoing a presentation about carbohydrate and our group member Alfira, Ammir, Ayu Vidya, Feri Andirawan, Kharisma, Mutiah Handayani, Mardiyah, Maylida Meilga Rosita, Monic Pamelia Puput Putri Ridwani and Rosita Nurfitrian. Now, next. Our topic today about Molly's test, Barford test, BL test, iodine test, Benedict test, acid mustard test, ozazone test, and Sally-1-Offs test. Next. Molly's test is performed from the percentage of carbohydrate that have more than 4 carbon atoms. In the test, the carbohydrate is treated with concentrated sulfuric acid, which results in its dehydration to produce an aldehyde. Subsequently, the aldehyde condenses with naphtol or a component of Molly's region to form a purple ring. In alkaline solution, the reducing sugar reacts with prussic acid, resulting in the reduction of prussic acid to preceramic acid. Prussic acid is yellow in color, while preceramic acid is red. The Benedict solution contains sodium citrate that prevents the deterioration of cupric ions to cuprous ion on storage.
Barford test. Barford test distinguishes between the monosaccharides and reducing disaccharides. In the acidic medium, cupric acetate is reduced to cuprous oxide or red PPT. Although Barford test is carried out to detect the presence of monosaccharides and reducing disaccharides, the time taken by disaccharides is more as they first get hydrolyzed in the acidic medium. The polysaccharides and oligosaccharides containing keto group get hydrolyzed due to the presence of concentrated acid and ultimately from furfural. This furfural reacts with resorcinol to product a deep cherry red color. The test can distinguish between aldose and ketose sugar as aldose sugar react at a much slower rate and produce a fine pink colored product. Next. Hexose sugars. In the presence of the acidic medium, the pentose sugars from furfural which subsequently condense with orosinol in the presence of ferric ion or but are present in pH region. To form a blue-green product, however, under the same condition, the hexose sugars from hydroxymethylfurfural and ultimately a brown-gray product is formed. This test can distinguish the sugars containing galactose when sugars react with an excess of phenylhydrazine, the formation of ozazone occurs. Ozazones are colored crystal and are easily detectable under a microscope.
Iodine test. Iodine test is chemical that distinguishes mono or disaccharides from containing polysaccharides like amylase, dextrin, and glycogen. This test has variation termed charge iodine test that is performed to indicate the presence of glucose may be placed in the levels. Principle of loading test. Number one, loading test is based on the fast the polydide in from color at adsorption complex with helical kinds of glucose result of amylase blue black, dextrin black, or glycogen red brown. Number one, monosaccharides, disaccharides, and barricade polysaccharide like glucose remain colors amylopectin procedure orange yellow. Number three, the reagent used in the iodine test is local iodine, which is an aqueous solutions of elemental iodine and potassium iodide. Number four, iodine on its own is insoluble in water at of potassium iodine result in a reversible reaction of the iodine ion with iodine to form a triiodide in ion which further reacts with a iodine molecule to form a pentadiene ion. Number five, Benz iodine solution upper brown, where the iodine, triiodine, and pentadiene ion are color. Sec, it is observation that the helicoil or spring structure of the glucose can is the key to this test. Number seven, for future the result color depends on the lack of the glucose can. And the triiodide and pentadiene ions format are linear and slip inside the helix structure.
Okay, next, Benedict test. Benedict test is a general test for aldehyde and alpha hydroxyl ketones. It can be used to detect the presence of reducing sugar in any given sample. Number two, the main contribution of Benedict region was the rapid detection of reducing sugar by color change using stable alkaline agents that were not very corrosive. Next. Principle. Carbohydrates with free aldehyde or ketones group have the ability to reduce solution of various metallic ions. Reducing sugar under alkaline condition tautomers and from enedials to enedials are powerful reducing agents. 3. They reduce cupric ions to corpus Cu2 plus from and are themselves convert to sugar acid. For the corpus ions combine with ohamine ions to form yellow corpus hydroxide, which upon heating is converted to red corpus oxides.
And the next is Benedict test. The procedure, number 1, add 1 milli of the 2% sugar solution to be tested to 5 ml of Benedict solution, or fresh solution can be prepared by mixing 10 gram sodium carbonate and 17.3 gram monosodium citrate dehydrated in inactive 5 milli water, and while stirring add 10 milli of water containing 173 gram copper sulfate, then make to a final volume of 100 milli. And number 2, place test tubes in a boiling water bed. Number 3, formation of red to brick red precipitate is a positive result. Next. This is a reference from Benedict test, and the carbohydrate are types of nutrient that has a function as the source of energy for a body. Benedict test is test used to test for the presence of reducing sugars. Alkaline copper solution, when reduced by carbohydrate that have free aldehyde of ketone groups, will form cuprous oxide, which are yellow to red in color.
The next purpose Benedict test aim to determine the presence of reducing sugar or compound capable of reducing aldehyde or ketone group in 1% glucose solution, 1% fructose solution, 1% sucrose solution, and 1% amylose solution using direct observation method. The result obtained were 1% glucose solution and 1% fructose solution showed positive result, while 1% sucrose solution and 1% amylose solution showed negative result. Next. The result of Benedict test can be used to test the content of reducing sugar in a solution. Alkaline copper solutions, when reduced by carbohydrates that have free aldehyde or ketones group, will form copper oxide, which is yellow to red. A 1% glucose solution and a 1% fructose solution yield positive Benedict test result because they are both monosaccharides or reducing sugar. One the reducing sugar group. 1% sucrose solution comes from the disaccharide group and 1% amylose solution comes from the polysaccharide group. Next.
And the next is conclusion. A 1% glucose solution and 1% fructose solution yield positive Benedict test result because they are both monosaccharides or reducing sugar, while the 1% sucrose solution and 1% amylose solution show negative result because they were not from the reducing sugar group. 1% sucrose solution come from the disaccharide group and 1% amylose solution come from the polysaccharide group. Next. And the next is acid mustard test. The mustard acid test aims to distinguish between glucose and galactose. The theoretical basis is that the oxidation of carbohydrate with concentrated nitric acid will produce soluble acid. However, lactose and galactose yield insoluble mamic acid. The tools used in the test experiment: test tube, dropper pipettes, water heaters, and microscope. The material used in the test experiment is sucrose, maltose, galactose, glucose, and concentrate HNO3. And the acid mustard test is the procedure. In the mustard acid test, the test solution was added with 2 drops of the concentrate HNO3 and heated in bath for 30 minutes in the cool, and the observed of the change that occurred. Then take glucose, maltose, sucrose, galactose under a microscope using an object glass and record the result. And number 3 in maltose, there are no spots. Sucrosa is the form of thick spot. Galactose is the form of strip and glucose in the form of spot. Number 4, so this can discuss glucose and galactose. Next, and the thing in the acid mustard test, and saccharide by a reaction with HNO3 or nitric acid. The result is of the oxidation of other monosaccharides dissolved in carboxyl acid, while the resulting galactose is insoluble in mastic acid. In the principle, the oxidation of the galactose with nitric acid carboxylic acid with is called mustard acid.
Osazone test. Osazone test is a simple test for the identification and qualitative determination of sugar. Sugar react with phenylhydrazine and from characteristic osazone crystal. For example, monosaccharides like fructose and glucose give the needle and broom-shaped osazone crystal. Objective of osazone. Number 1, to detect reduction sugar. Number 2, to difference reduction sugar from non-reducing sugar. Number 3, to distinguish different reducing sugar between each others.
Gram of phenylhydrazine hydrochloride and 0.1 gram of sodium acid. Number 2, glacial acetic acid. Number 3, test sample. Procedure take 5 millimeter of test solution in a clean, dry test tube. Number 2, 0.3 of glacial acetic the test tube. Number 3, mix it well and warm the test tube gently in the water, but if required to dissolve all the elements. Number 4, keep the boiling water and observe the formation of crystal at various time points. Number 5, observe the set of the crystal under low magnification under a microscope. Next.
Osazone test. Principle of Osazone test. Number 1, the reagent for this test consists of phenylhydrazine in acetate buffer. Number 2, this test is based on the fact that carbohydrate with free or potentially free carbonyl group react with phenylhydrazine to form acetone. Number 3, the condensation, oxidation, condensation reaction between 3 molecules of phenylhydrazine and carbon 1 and 2 of aldose and ketose yield 1, 2 diphenhydrazine, which is known as acetone. Number 4, since both carbon 1 and 2 are involved and the two epimers produce the same osazone. Number 5, similarly ketose with a configuration identical to aldose below, see glucosa and fructose.
Result and interpretation of ozone test. Based on the shape and structure of the crystal and their time of occurrence, different sugar can be identified. The of the reduction sugar through this test. The present study was formed on 6 monosaccharide, glucose, fructose, and 2 disaccharide, is maltose, lactose. To solve the stable different sugar can be identified on the basis of the structure of the crystal and the time of occurrence. Is This is a tool of the observation from a microscope. When observation model crystal were formed after a specific time interval, either by boiling or by cooling solely after boiling, when examining under microscope, characteristic crystal were found to make of one picture is nailing seven crystal is and and uh, picture the second picture another set crystal is and picture crystal manusia and for me set crystal is galactose and five uh, when the nebula set crystal is silos and the last one uh, okay next Celiwana of test resulting of that is used to this of cancer, glucose or other negative result of cherry red. But if heating is continue for more than 5 minutes, those will sometimes produce a red color young red. First, dissolve 0.1 gram of resorcinol in 33 milliliter of concentrated dihydroic acid HCl and make up the final volume 100 milliliter. Second, and 1 milliliter of 1% sugar solution to be test 10 milliliter of reagent. And third, heat the solution in a boiling water bath depth cherry or red color in 5 minutes so the pressure of ketosis as a figure. And the last one, sucrosa give a positive ketose test because partial hydrolysis to glucose and fructose. Next.
I will continue. Next is Seliwanov test, determination of total fructose content using the Seliwanov test. Fructose whereas it can reach with PMP region may be due to lack of aldehyde group on molecule fructose, which is an important group for the synthesis of PMP derivative. And the Seliwanov test has been shown to be efficient for the examination of fructose. Next. And this is for the various reference that we use. Next. and can what for sepurane aku lali